the first presentation emphasized the importance of the multidisciplinary teamwork in interpreting particularly variants of unknown significance, whereas the other two presentations, gave us an excellent idea about all the progress that has been made in gene editing and gene therapy. I think that we have with us in the panel the two commentators also, Dr. Gourzi and Dr. Pantou, who, if they have a comment, they can go ahead. And if I may ask the first question, is I saw in a report in Nature magazine last December, and it was like a promotion of biotechnology company who has a different approach, in gene editing. I think it was for the MYBPC3 gene mutations, where the chairman and the co-founder states that cardiology is still in the stone age of as far as gene editing compared, for instance, with immuno-oncology. Of course, we didn't see something like that today, but I would like to hear your comments about that.
Maybe I start, and maybe
yeah, Professor Karia, yes.
Yigal, you want to say something after? Well, I don't know which company contacted you, but I mean, there's different companies who are interested.
No, it was in Nature magazine. It was stated, by the co-founder of the company.
Okay, so there are companies, they are interested now to treat severe form of cardiomyopathy associated with myosin-binding protein C3, and one of them is Danacor in Switzerland.
And they are
going to promote gene therapy with overexpression of the myosin-binding protein C cDNA in the patients. So we have proof of concept in mice, proof of concept in iPSC-derived cardiomyocytes. It was not my topic today, so I did not present this, but I think they will make it because in these infants with double mutation, B allelic mutations, in myosin-binding protein C, and particularly if they are truncating, then there is no myosin-binding protein C. So this is the right strategy to go and to overexpress myosin-binding protein C. For the gene editing, we are not yet there, except I showed you the data of Eric Olson for Duchenne muscular dystrophy, but several groups are working in this field, and there will be a future of gene editing in cardiomyocytes in vivo in the next, I would say, 10 years.
Yeah, I'd be happy to add to that. I understand where, I guess I understand a little bit why it's said that we're in the Stone Age in terms of cardiac therapies, because I think we're not in the Stone Age in terms of understanding, because I think the previous speakers, like also Dr. Van Tintelen, Professor Van Tintelen and Professor Correa illustrated, there's a huge advancement in understanding and designing the right molecules for gene therapy is also done, I think, very proficiently. It's just the delivery. I think the cardiac tissue is a very tough one to target. And on the other hand, that means that we can jump from Stone Age to, let's say, science fiction as soon as delivery gets improved. And I think it's going to be Star Trek. So it's from Stone Age to Star Trek in a couple of years.
And if I may add something, there is a big, I don't know if it's only in the cardiomyopathy field, I think it's in other areas also. There is a big gap between the laboratory which is going to recognize the genetic defect, which is going to design the therapy, and until this therapy reaches, the patient. There are a lot of experiments to be made. If this therapy is tailor-made, like Professor Tinto has presented, it is going to be a therapy for one person or for one family or for a few families. So all the experiments on the toxicity, efficacy, all the paperwork that has to be done, no pharma company is going to pay this bill because it doesn't represent profit for them. So there is a big question on who is going to pay for this. gap. And I think in the Netherlands you have formed some kind of consortium for the founder mutation in the phospholabad. I don't know if it is EU grant, it is EU funded or nationally. I don't know. Maybe you can share how did you do this, because we have a similar, phospholabad founder mutation in Greece, and I don't know what to do next.
If I may comment to that, so basically you're telling, mutations or specific variants are rare, but on the other hand, if you take the bigger picture, they might not be so rare if you take the global perspective. And I think phospholamban is a great example. It's highly prevalent in the Netherlands, of course, but it's also present in Greece. It has been identified in the United States, in Canada, Norway, Russia, Italy, Vietnam, China, and I've probably forgotten a few other countries. So I think, and that holds true for many variants, the total number of cases globally might add up substantially. But I agree, there's always a huge gap between, from a single family to a well, probably expensive therapy. But maybe Yigal or Lucy can give some more insight on that part.
Yeah, I'd be happy to comment. I think it touches on a vital point that with advancing technologies, they get more specific, and then you bump into this problem because they get so specific that it's indeed, as you would say, you would ask for a whole trajectory, multi-millions per family, which nobody will do. So there's two ways about it, trying to use the fact that it's a generic kind of backbone structure, where at the end of the day, like with the COVID vaccines, we would change a nucleotide sequence, and there is already, for instance, there are discussions within the FDA about updates of the COVID vaccines that you don't need to go through all the studies again if you only change the nucleotide sequence of the mRNA vaccine. That could help us in this field as well, that if we just change the nucleotide sequence and the rest of the therapy is exactly the same, it could be kind of shortcutting a lot of the toxicity and safety kind of steps. So, and the second pathway, and Professor Elliott alludes to that, I think also, is that it needs more active, more proactive discussion from our side as physician scientists to approach the legislative authorities to see how we redesign the administrative issues around this type of therapies.
I think I don't have too much to add on this, but from my point of view, I think it's important to prioritize the groups, and there is different company working for rare diseases and severe forms, and I think this would be the way for gene editing or gene therapy in general first. I mean, the severe case should be treated as soon as possible, of course, with the consent of the family, and then for the less. So I see Perry Elliott who asked a question peu à peu. Who should set the priorities? That's a good question. I think it's open. I think it's a good meeting to discuss about this. I think it's also a teamwork between the family, the cardiologist, the geneticist, and the molecular biologist, together with the companies who can drive gene therapy towards the patients. And of course, the regulatory agencies. Thank you.
If I may just go back to the first talk, because we have some questions from the audience for Professor Van Tinkelen.
We cannot hear you very well. We cannot hear you very well.
I don't know exactly why.
Maybe the microphone is too close.
Yeah, don't have anything else open. So I think now it's working.
Yeah.
So, Professor Van Tinkelen, we have a couple of questions from the audience. So one is, in your experience, what percentage of VUS change classification occurs after the clinical input and segregation data. And maybe I can also give you this second one, which is what you think is the input that artificial intelligence can contribute to this in the future. And maybe a third one from me related to these two, which is from a very practical and clinical perspective point of view, which also alludes to the title of your presentation. What do you think is the best system to revisit the classification of these VOSs from time to time? There's probably not one single answer to this, but is it more the laboratories? Is it the genomic networks or hubs? Is it the honors more on the clinician or cardiologist side? I'd like to hear some thoughts about this as well.
Yeah, thank you very much. And well, thank the audience for so many questions. First of all, the reclassification, making use of segregational data. To our experience, roughly 10 to 15% of variants are being reclassified after we have studied segregation in a family. So 10-15% doesn't seem that much, but on the other hand, it makes a huge difference for a patient and his or her family. So, you know, I think every single case counts. So it definitely makes a difference. On the other hand, reclassification of a VUS or a variant is basically an ongoing thing, and that touches upon your third question. Who is responsible for the reclassification? And I think the system should be organized in a way that the big genetic laboratories should be responsible for this reclassification. So basically, it's bioinformatics. So input has to be generated from literature, databases, large registries. You know, for example, the Greece one that was mentioned yesterday, and also in the Netherlands and many other countries, we do have those large registries. So input from literature, input from large patient registries, input from control populations will definitely help to reclassify variants, and that should be done over and over again. And again, it's a dynamic process, and it has to happen, you know, every time additional information is available. So I think it's an ongoing thing.
Thank you very much. I think
Professor
Elliott just introduced another question for you, which is more the gene-based perspective, the GUS question, rather than the VUS.
So basically, the question is, you know, is a gene related with disease? So basically, if you start with a patient and you do a genetic analysis, there's of course, is there a relation of that specific gene to a specific disease? That's step one. And the second step, like I discussed in my presentation, is of course the interpretation of the variant. And to my opinion, those gene studies are important as well, because we have definitely to keep in mind that there's always this kind of, some call it background noise. So specific variants in specific genes are present in the general population as well. You know, I think a great example are PKP2 mutations in the general population, which are present in roughly 1 in 400 people. They carry a truncating PKP2 mutations, and they are well known for their relationship with arrhythmogenic right ventricular cardiomyopathy. But it's important to keep in mind that there's also this issue of penetrance. So not every mutation leads to disease in a specific person, and we still have to learn a lot about it as well. So Perry worries about studies that show that some genes are associated with disease or are not associated with disease. I think information about genes is important as well, like it is for specific variants.
I think that we have to be mindful, obviously, that there's a whole big bunch of literature lately with analyzing statistical difference between cases and controls with aggregates of rare variants in a gene. And perhaps we have to be careful about broad statements that a gene is or isn't associated with a certain condition, because then we have exceptions. And I think a good one is alpha-actinin, that Professor Carrier just referred to, one specific mutations, and there are other examples of missense variants that are definitely doing something and pathogenic. But if we look at actinin, I think in those type of statistical comparison papers and curation papers, alpha-actinin has always been down in terms of being pathogenic for HCM. So individual mutations definitely are important as well.
Yeah, and sure, and I think the example that was given also underscores the importance of those functional analyses. So I
Absolutely.
think that's a great addition.
So I'm aware we have another discussant, Dr. Gourzi, which I think still didn't have an opportunity to comment. And before we finish,
good afternoon to everyone. Thank you for these very interesting presentations. Actually, I have questions for Dr. Carrier and Dr. Pinto, who is, I think, left, I guess. Anyway, I would like to ask what limitations should be addressed for the efficient and safe clinical translation of this technology, of CRISPR technology, for silencing RNA in the clinical practice. And I know that also many researchers argue that all this system, like mice specifically, do not faithfully recapitulate the presentation of cardiovascular diseases in humans. So how you're going to overcome these problems and proceed to human studies?
Yeah, so thank you very much for the question. Well, I mean, if you want to bring a gene therapy medicinal product to the patient, you have to move from mouse to human, right? So you need larger animal models to test the therapy. And so, for example, the non-human primates or the pig models. And also, you need to evaluate your therapy in a human context. And what I presented you today with the iPSC-derived engineered heart tissue could be a good platform to evaluate the different strategies in a more automatic way. Now the limitations, of course. I mean, first, we need to have the gene therapy medicinal product at the right place, meaning in cardiomyocytes. So you need to have a specific delivery. We need to have a specific adeno-associated vector with a cardiotropic effect. And we need, I mean, either for gene editing with CRISPR-Cas or gene therapy with overexpression. And you need also a specific cardiomyocyte-specific promoter to express the Cas9 only in the cardiomyocytes. If not, I mean, Cas9 can cut everywhere, right? So you have to revalidate before going to the patient that this is not the case. But there is a lot of new developments of gene editing. I did not enter the detail today, which will be pursued, like prime editing and other technology, which will allow to precisely cut at the right place without any side effects. So these are and finally the limitation. I mean, the time. I mean, you have to test. You have to, of course, as Mrs. said that we need to validate all the toxicity of the compounds, and this has to be done it can be done in mice. And of course, the mice do not reproduce all the phenotype, but in the case of cardiomyopathy, I think the mouse models had the heterozygous state. In general, did not reproduce the phenotype of the patients, and you need to have two mutations to reproduce human HCM, for example, phenotype. So there's some limitation in all the models, and I think the next years will give us more emphasis about the developments.
Actually, I have a technical question for Dr. Van Tintelen. So, do you believe that we should, as a clinical geneticist, should we use small panels in order to perform genetic testing for cardiomyopathies, or should we use whole exome sequencing or whole genome sequencing?
Yeah, so again, what I emphasized at the end of my presentation was interpretation is teamwork. So it's highly dependent on your team and the people that are present in your team, whether they can handle the information that comes back from whole genome sequencing or whole exome sequencing. So I think, if you do not have a very experienced team around you, there's nothing wrong with basically making a panel smaller. And for example, a cardiomyopathy gene panel can be whole exome based. So basically just filtering out the known cardiomyopathy genes. And it also gives you the opportunity to reevaluate the data at a later time if new genes come up and pop up. So again, I think it's highly dependent on the team you're working in. So to give you an impression, what we currently do in diagnostics is we use a exome-based gene panel, and nowadays we move on to a whole genome-based gene panel, but we filter out the many genes that have nothing to do with cardiac disease.
Thank you.
So we need to conclude. On behalf of my co-chairs, Drs. Lopes and Degiannis, and the coordinators of the discussion, Drs. Gourzi and Pantou, I would like to thank the speakers for their excellent talks. It was a very, very interesting session on the novel molecular methods and findings, and the new era of precision medicine that we're heading to. There are many challenges that we need to discuss about. This was a first nice discussion, but there are issues that come up, like administration issues that were mentioned, or economic issues, and of course, legal and ethics issues. The discussion is, I think, now open, and we need to address all these issues, also in the context of networks like the GRI Cardiac Network in Greece, that we're very proud of and very happy to participate in. Thank you all for attending. 2 minutes break will follow before the next session. Thank you.